discuss about the transport across cell membrane, and there are various transport which are occurring without membrane. Do you know such system? So such transport are called as diffusion. You can see the perfume when you are spraying, a lot of molecules are coming out of the bottle and these are in high concentration, right? So they are diffusing till they achieve equilibrium. Do you know equilibrium? Means equal concentration. Equilibrium, equal concentration. So now why transport across cell membrane is required? So you might be knowing about the membrane structure, right? And this is made up of phospholipid component, which is phosphate here, okay? And the lipid portion is also there, okay? So this is hydrophobic and phosphate portion is hydrophilic, okay? And now what you can see, there are integral proteins. This one is integral proteins, okay? So they are facilitating diffusion like ion molecules or larger molecules which cannot cross the membrane. So they are being transported by which diffusion? Facilitated diffusion, right? And also if you see, there are a lot of receptors containing the sugars, right? Sugars, okay? So protein plus sugar, they are forming sugar is forming a kind of receptor, right? And if you are talking about the transport across cell membrane, we must not forget about the osmosis. Osmosis is the reason how the root is taking water from the soils and they are reaching to the top, okay? So let us discuss, let us begin. Myself again, Dr. S.M. Bhatt, right? So we are going to discuss what? I'm going to discuss you a lot of things today, okay? So I'm going to discuss a lot of things today with you. For example, if you are spraying the spray, then there is a diffusion, how the water is transported after the absorption from the soil, right? Because of water potential difference we are going to discuss, and there is a voltage gated channel, right? Voltage gated channel, which are channel protein, which is the part of facilitated transport, right? And you can see there is a, why the name is voltage gated, because voltage is going to change because of this release of these neurotransmitter from receiving end to, this is the receiving end, this is the sending end, right? And there are various other examples, and if you'll see, various molecules need to be transported. For example, water, right? Water, if going from inside to outside, this requires a special class of protein, channel protein called as aquaporine. Do you know about the aquaporine? Aquaporins. Porins, right? Aquaporins. P-O-R-I-N-S. So now how the gases are going to diffuse? You know, they don't need any kind of membrane protein or channel protein or carrier protein. Directly they can diffuse according to the concentration gradient. Glucose, because we are eating a lot of food, so everything is going to be converted into the glucose. So they must be diffused from either inside to outside or outside to inside via this facilitated diffusion mode, okay? So I will tell you later on, facilitated diffusion occurring via two modes, either carrier protein or channel protein. Carrier protein and channel protein, if you talk about the differences, so you can identify easily, because carrier protein is going to change the shape, but here you cannot see any change in the shape. So obviously this is a not carrier protein. This is a channel protein. Okay, and similarly ion is also going to be transported across the membrane via this channel protein. And according to the name of ion, for example, potassium is if going to cross from inside to outside or outside to inside, so they are going to call potassium pump. If sodium is going to transport, they will be called a sodium pump. So similarly, according to the name of ion, the name of channel protein or carrier protein you can name, right? For example, if a steroid has to be passed, you can see they are directly passing from the membrane. They don't need any kind of directly passing, direct transport, right? What you can say, this is a kind of direct transport. Why? Because inner is hydrophobic. Inner core is hydrophobic, correct? And steroid is what? This is also hydrophobic. So similar mix with similar, they are going to mix well, and that's why they dissolve. For example, you are applying the drug, you are applying the oil, all they are the steroids. They easily can directly be transported. So in today's lecture, we are going to study a lot of peculiar things. And heart attack because of cardiac arrest, because of potassium pump, because of sodium pump. So because ATP is involved, ATPase is involved, so that's why this is called as sodium potassium pump. Okay, sodium potassium pump. So pump is responsible for formation of ATP. Do you know that? And this ATP formation via either V ATPase or F ATPase, this is a kind of motor. Okay, have you seen the electricity production by the water pump? Right, water falling from very high location over this machine, and this machine is moving round and round and round. That's why a lot of turbine is moving, and turbine producing the electricity. Similarly, the ATP is produced when there is a proton pump. You can see in the VATPase only 2 proton is going to be pumped. In the FATPase, this 4 proton is going to be pumped directly, right? So that's why you can see there is a combination of ATP plus PI, that's called as ATP formation, right? Here you can say ATP formation by combining the ATP plus PI. This is breaking of the ATP. This is kind of breaking of ATP. Why? In FATPase you can see there is a ATP production is going on. Okay, so I'm telling you why the importance of transport. Ion channel ion pump, you can see here active transport is there because ATP has been consumed in transport of 2 potassium inside and 3 sodium outside. That's why this is called as sodium potassium pump. And this is creating a negative potential inside the membrane and positive potential outside, and that is essential for the polarization of the membrane, right? So you might be heard in the neurotransmission. And a very important part is that how the glucose is going to be transported inside and across the membrane. Glycerol pump is there. Drug I have shown you directly they can be transported. So if you'll see about the facilitated diffusion, this is very commonly asked term in the examination, facilitated diffusion. So this facilitated diffusion involving the carrier protein. How you can differentiate? You can see this is structure carrier protein, and this is the channel protein. What is the basic difference? You can see this as opened. So now here what you can see here, this is opening inside cytoplasmic side, and here this is opening toward the extracellular side. Means this channel protein, this is what? This is channel protein, they are going to change the shape. Okay, they are going to change the shape. So already we have discussed how in exchange of sodium, glucose is transported. This is called as a secondary active transport. Primary active transport, sodium plus pump moves sodium using the energy of ATP hydrolysis to establish a concentration gradient of sodium. Why this is called a secondary active transport? Because they derive the transport of glucose against the concentration gradient with the help of sodium, which is moving along with the concentration gradient. There are various pumps also available in the membrane called as antiporter. You can see the one molecule, red one is going towards the extracellular fluid, while blue one is moving toward the intracellular fluid. So they are moving in anti-direction, opposite direction. What about the co-transport? So if the sugar molecule is moving, one sugar molecule is moving with the one sodium molecule, this is called as co-transport or coupled transport, because without movement of sodium, no sugar will be transported inside the cells. So again you can see a second example of secondary active transport, right? Symport in the same direction, antiport in the opposite direction. How the diarrhea is going to occur? This is because of disturbance in, you can see, which protein? Carrier protein, right? Which disturb because of infection of toxins, bacteria, or certain viruses, right? So we are going to treat with the ORS goals. Okay. So now you can see, we can classify the third type of bulk transport. You can see there are three types of endocytosis. One, when the solid particle is being eaten by the cells and the membrane is involved in forming the vesicles, food vacuoles, then this is called as a phagocytosis. Pinocytosis, when liquid is going to be packed into the membrane vesicle, and when the receptor is involved in catching the antigens, right, and then identification of those antigens, and they are coated by a protein, special protein that is called as the cladrines. So we will study that. It's also called as a receptor. Now you can see here receptor mediated cytosis. This best example is cladrine coated vesicles, because cladrine can be rearranged again and again. They are going to be recycled. They are not going to be formed again and again. Right? Otherwise, there will be lots of energy. And what is cladrine? You can see cladrine triskeleton structure have been shown, which are having the heavy chain and light chain. So they are forming the cladrine coat. Right? Dynamine is there, and if you will see, there are cladrine coating. This is a cladrine coating. Okay. So which are packing very effortlessly, right? So we are going to discuss one by one. Just a minute. Again I came back. Hello students, hello bachcho, how are you? Again I am your mentor, Bhatt sir, again with you. Now you can see the membrane is there. What kind of, how molecule can go across the membrane? So passive diffusion is one, and then facilitated diffusion, second one, with the help of certain channel protein or carrier protein, and then you can see the selective facilitated diffusion, And then fourth one is primary active transport, where ATP is involved, right? And then secondary active transport, you can see symport and antiport. I have given the example because in exchange with the other molecule, some molecules are going across the membrane. For example, glucose can be transported in exchange with the sodium in symport direction, same directions, right? So bulk transport, if we are talking, there are two types: endocytosis and exocytosis. So let us learn one by one. Okay, so what are the bulk transport? Before going into that detail, we should again give a brief classification on the transport. There are two forms of transport: passive transport, active transport. Okay, so diffusion, osmosis, you can see here, you can classify them because they are not involving any energy. No membrane is there, but here is the membrane is involved. Because they are moving from high concentration to low concentration across the semi-permeable membrane, that's why they don't need any energy. Okay, while if you are talking about the active transport, there are two types: phagocytosis, eating by cells like phagocytes, amoeba, macrophages; pinocytosis, drinking of cells; and then third one we have discussed about receptor mediated endocytosis, right? So cell transport, again you can see the classification: passive and active, and then passive, for example, simple diffusion, osmosis, facilitated diffusion, three type. And active we're talking pump vesicles, right? A pump can be various type and vesicle based, that is also called as the bulk transport, where vesicle is going to be formed essentially in exocytosis, endocytosis, right? So if you see, endocytosis can be classified into three: pinocytosis, phagocytosis, receptor mediated endocytosis, right? And a pump can be primary, secondary, right? And exocytosis, right? Means this is reverse of endocytosis. Endocytosis, anything coming inside the cells. Exocytosis, when anything going outside the cells. Okay, so exocytosis, breastfeeding by the babies, right? Mother's milk coming out of the breast because of exocytosis. Hormones going to be secreted by exocytosis. Digestive enzyme going to be secreted by exocytosis. Already we have discussed about the hormones and breast milk, right? So all these are very good example of exocytosis, right? So simple diffusion from where solute molecule generally not moving in any kind of diffusion except free diffusion. If you're talking about the free diffusion, simple diffusion, then solute moving from high to low concentration, for example gases, right? And if you're talking about what difference between diffusion and osmosis, here you can see solute molecule are moving from high to low concentration. In osmosis, there is a semi-permeable membrane and only the solvent molecule. Note that solvent molecule is moving from low to high concentration and the membrane is semi-permeable. Yes, you are right. So solute is moving. Solute, for example, sugar, if dissolved in water, okay, so sugar molecule will be solute and water will be solvent, okay. So here solvent is moving, water is moving, right? And here in simple diffusion, sugar is moving, okay. So what about the idea of water movement? How to decide how the water moving in plants? Okay, so this is a general formula. So water have the highest water potential. Okay, highest water potential. So if anything, if you are adding, for example, if you are adding the salt, if you are adding the sugar, you are adding the organic substance, vitamins, and everything is making the water potential psi negative, which is less than the water. So easily you can define, if anything in moving so water to less W, right, less W. And if there are two substances, for example A to B, and they are differential by permeable membrane, then where to where they are going to move, you can see the water potential. If water is high, so water potential automatically high over sign, right? You can show the water potential by this sign. So this will be the high, this will be low, because some solute is dissolved. So obviously the movement will be from A to B. And this system is the decisive factor for hypertonic solution or hypotonic solution and their movement. For example, more water is there, then hypotonic, right? And less water, more solute is there, this is hypertonic. So less water, then obviously from more water to less water, hypotonic to hypertonic. What is moving? Water, solvent. So solvent move कर रहा है, so that's why this is called as a what? Solvent is moving, osmosis. Across the permeable membrane is there, the osmosis. So osmosis, you can see the different condition. If medium is high in water, hypotonic, low in water, hypertonic, and same in water, then isotonic. Isotonic, you can see there will be no bursting of the RBC when the inside and outside environment is same. Isotonic solution, right? Isotonic solution. You can see here isotonic solution. No net gain, loss of water shall remain the same. And if animal cell is placed in hypotonic solution, then what is happening? Hypotonic, okay? Water is more outside. Okay, so then water enters inside the cell and the cell is going to burst. So in each condition, cell is going to burst, this condition. And this is the flaccid cell shrink case, because outside water concentration is very low. So from inner cytoplasmic to outside, the water is moving completely. Okay, so if isotonic solution is there, there will be low movement. Hypotonic solution, cell shrink. Hypotonic solution, then bursting will occur. Now what you can see, how to decide the movement of water. So pure water have this maximum water potential, right? And if you are dissolving anything, then psi will be have the negative number. So obviously the direction of movement will be from this maximum number to negative number. You got the point? So what official you can decide the direction of water movement based on calculation of water potential. So this is also helpful in deciding the solid potential, pressure potential, and many things we will discuss later on in the other chapters. Okay. So now this is homework for you. Which chamber has lower water potential? Which chamber has lower solid potential? In which direction will osmosis occur? And if one chamber has psi of minus 2000 kPa, either is 1000 kPa, which is the chamber that has the higher psi? So higher psi means less negative. So less negative is this one. Okay. So less one negative means this is water potential is high. So they are asking has higher water potential. So 1000 negative 1000 Pascal will be the decisive factor. Okay. So passive diffusion, again we have discussed there are two types. Facilitated diffusion means there are certain channel protein or carrier protein, and simple diffusion directly they are moving, and Already we had discussed about active transport are of two types: primary, secondary. Primary uniport for transport, pump mediated. Okay, right here. Okay, so what I'm saying, secondary transport, antiport, symport, carrier mediated. Pump mediated, this is uniport, co-transport. Already we have discussed. Passive transport, already we have discussed. Single direction is there, high to low. So uniport. Simple diffusion, high to low. That's why this is also free diffusion. Channel mediated, carrier mediated. Both are example of uniport facilitated diffusion. Okay, so in next topic we are going to discuss about certain more things like what is the difference between channel protein, carrier proteins. You must tell me in the comment box what is the difference between channel protein, carrier protein, how the voltage gated channel going to work, and what are the aquaporins, what is their role, where they are going to be conserved in bacteria, plant or animals or not. And already we have discussed about the osmosis, and then water movement, active transport, and in the next class we are going to discuss bulk transport. Thank you so much. Bye bye. Have a nice time. Keep studying. सब्सक्राइब कर लीजिए हमारे चैनल को, लाइक कर दीजिए, कोई प्रॉब्लम है कमेंट कर दीजिए. ऐसे रिसर्च बेस मैं आपको बनाऊंगा, इंटरेस्टिंग वीडियो बनाऊंगा. दिस इज़ माय कमिटमेंट. This is my commitment for NEET, my commitment for NET CSIR. This is my commitment for the 12th class, ICC board, CBSE board. Okay, तो सारे लोग देखो, सारे लोग सुनो, सारे लोग समझो इस बात को. Right? And call me or you can join my channel. Okay. So if you want my WhatsApp number, I can give you also in the comment box. I'm waiting for your comments. Okay. Thank you so much. Okay, bye bye.